Hello and welcome to your first set of Bio43 pre-lab videos. I'm Professor Brown, one of the Bio43 instructors and the lead architect for this virtual lab curriculum. When faced with the concept of running a remote lab, we thought the best course of action would be to put you in the role of a principal investigator, or PI. The PI rarely performs the experiment themselves, relying on assistants to collect the raw data. The PI is essentially the head of a research lab. They are the person responsible for deciding what questions to investigate, which hypotheses to test, how to test them with the right experiment, and how results should be analyzed and reported. So that's the role we're giving you. You will formulate interesting and good biological questions and hypotheses to investigate, and design experiments that your instructors will carry out. After the instructors collect the data, they'll give it to you to analyze, decide if you've made part of a new discovery, and write up a report. In other words, you'll do 90% of what a senior scientist does, everything but the data collection. To decide which questions you want to address in your lab, you have to be knowledgeable about the state of the field. In the first week of lab, we'll introduce you to the two major types of articles you'll find in the scientific literature: research articles and reviews. A research article, or primary literature article, is a direct report from the scientists who performed the research. These articles are very similar to the reports you'll be writing in this course, though at a larger scale, featuring the results of months or even years of experiments. But they follow the same format, with introduction, methods, results, and discussion sections. Often this writing is targeted for people already familiar with the field and can be difficult for newcomers to parse. So this is where we'll dedicate the most time in the first week of lab. Review articles have a broader scope. The writers look at dozens of primary literature articles on a given topic and summarize the most important findings to provide an overview of the state of the field. A review won't have the same sections as a research article, since it's essentially one long introduction to the field, perhaps with some broad conclusions at the end. Most notably, there is no methods section, since no new experiments are performed. A review article is typically written to be understandable to scientists outside of the field, and is not all that different from reading a chapter in a textbook. In summary, a review article is a good entry point for learning about a topic, but research articles give you the most cutting-edge information and show you details of how experiments are performed and how data is analyzed in that field. There are other types of articles you'll come across when performing a search of the scientific literature, the focus in our 5th week of lab, but we won't use them in this course. The focus in our first unit will be cilia, hair-like structures that can be found on most types of human cells. Motile cilia make a sweeping motion against the extracellular fluid, causing either the movement of the cell or the circulation of the fluid. Such cilia play important roles in development, fertility, and prevention of respiratory infections. Of course, we can't study cilia in living people, and even human cell lines have severe limitations. Tissue culture work is expensive, classic genetics can't be done since you can't mate individual cells in culture, and most human cell types have very few cilia, making it difficult to get enough for large-scale biochemical experiments. Because of this, we turn to model systems, non-human organisms that can be easily and ethically studied in the lab, yet are similar enough to us that our discoveries are likely to apply to human biology as well. In this lab, we will use Tetrahymena thermophila, a single-cell eukaryote that can be found in freshwater ponds. You can even find some in Fake Lake at the Bernard Field Station. The evolutionary divergence of humans and Tetrahymena was about 1.5 billion years ago, but cilia are so important that much has been conserved across the years. If we find something that affects cilia function in the Tetrahymena in our lab, it's likely that thing will affect the cilia in our lungs as well. In week 1, you'll learn how to gather the scientific background on cilia and Tetrahymena that will later be necessary for developing a hypothesis. On the lessons page for this week, you'll see links to a review article and research article on this topic. We'll go through them in class, but browse over them quickly and note the difference in structure between the types of articles. In week 2, we'll walk you through a sample experiment and data analysis. Then in week 3, you'll develop your own novel research question and hypothesis and design an experiment to test it based on what you've seen. I hope this video has given you an understanding of the major types of articles you'll find in the scientific literature, and provided a good introduction to what it will be like as a PI of this lab. The next videos will go over the basics of experimental design and hypothesis testing to help you in this role. See you next time!